So welcome to TRICEP. This is a new facility that we've created in order to take the fundamental advances in material science and engineering into a manufacturable prototype or a commercial product. You've seen all of the fundamental work that we've done, for example, with graphene and other biomaterials, what we've been doing in terms of processing cells for 3D bioprinting. And you've also heard about some of the practical applications where we can use that fundamental knowledge. Here we focus on those practical applications and we focus on developing protocols for the bioinks that make them ready for manufacture. We focus on developing the protocols for the customized printers that make them ready for manufacture. This is an exciting new development, and we're going to have a look at what we're doing in those areas of bioinks and bioprinting and how we're addressing some really critical medical challenges by creating that commercial opportunity. So we'll go and have a look at TRICEP. This is a great facility incorporating all of the bioprinties, development of new bioprinters using conventional 3D printing, but also, as you won't find in a conventional university research environment, taking all of those processes and making them ready for manufacture by implementing a quality management system that allows us to realise real commercial opportunities. I'm going to have a chat with Alex about what he's been doing with bioinks. Alex works here at TRICEP. He's an employee of ANFF, the Australian National Fabrication Facility. So, Alex, you get the fundamental formulation of the bioink from our colleagues across the road at IPRI and ACES. And so what challenges then do you have in terms of really scaling that up and turning it into a reliable product?
So everything with upscaling becomes a lot different. Every parameter of these experiments and the results that we get out have to change. So what we have to do is really explore every single parameter in these experiments piece by piece so that when we are making larger batches of these bioinks, they'll be, as you said, reliable and the same. So there are a lot of challenges, but it's a slow working process.
And so I guess we also really need to go right back to the source of the materials, don't we?
Yeah.
I mean, for some of the research work that we're doing, of course, getting small amounts of material in order to do those small scale experiments is relatively easy to achieve. But here you really need to know what's the primary source of those materials in order to ensure quality control. Is that correct?
Yeah, that's correct. So we have a really nice network of collaborations around UOW and TRICEP. The one that comes to mind is a lady called Pia Winberg. She is our main collaborator in producing us with a type of seaweed called ulvan, and we here are making bioinks from ulvan. And it's a part of that nice collaboration that also helps us know that we have a high standard. So before we start making any large-scale production of the bioinks, we have to undergo a suite of characterization to know that they're high purity and then, a very good product for us to work with.
So you would characterize that material on arrival to ensure its quality, and then you would start to do whatever reactions you need to do to turn it into the bioink. Could we have a look at one of the reactors you would typically use?
So we start off at these smaller scales. This is a good example of a reaction that we would particularly do over at AIM. And so taking the results that we would make from this, we would hope to achieve similar results in the large-scale reactors. So I'll take you over to the...
So what sort of quantity of material would you be generating from a reaction of this scale?
So in this scale, I would say you could obtain 20 to 50 grams is the scale.
Okay, so that's still quite large scale compared to what they would do in the research facility, right?
Yeah, definitely.
So it's one step up.
Yeah, research is about 1 gram. We're working towards 50 grams here. Right. And now I'll take you to the reactors. These reactors are state of the art. So these reactors, this is a 10-litre reactor. This is capable of generating quantities of up to 500 grams. All of this has been controlled by a thermoregulation unit, which is plumbed through the external walls. We can control the temperature extremely sensitively. We can collect our material from the bottom, which means everything remains extremely consistent when comparing it from the small scale to this larger scale.
Yeah.
And over here, we go up a little bit further, and this is our 20-litre reactor. And this 20-litre reactor is capable of producing kilogram batches. Kilogram batches is the, in my opinion, the big leap into an industrial process.
Right. And so Sanjeev was highlighting to us when we were talking about the scalability of graphene processing, the importance of simplicity. So what are your thoughts on that?
I agree. This is as simple as it can get without reinventing the wheel. This has a simple heater and a jacket to keep it warm with just a big motor to stir it. It's as simple as it can get.
Yeah, and I guess it's important that the actual chemistries used are simple and is the minimum number of steps possible. Right?
Exactly. So in the work that we've done here, we've managed to reduce steps significantly from the lab scale. So from the 1 or 2 grams, we're able to reduce those 10 steps into 2 or 3. Right. And that's a big part of process chemistry.
Yeah, and that's a huge step forward, getting down to 2 or 3 steps which you need to control accurately. And I guess once you have that material, then you've characterised what went in, and then I guess you need to characterise what comes out.
And right? So the first step in doing that process is to purify it. So on a kilogram scale, this is our new piece of equipment that we have recently customised. This is a cross-flow dialysis purification system.
Wow.
This system is capable of running through about 140 litres of our reaction mixture. It'll pass through a membrane that sits in here, which works via dialysis, and it will throw away any side products and impurities that we don't want, and leave us with the pure material in this batch.
And so compared to processes that would have been used before, what sort of time saving are we talking about here using the cross?
So on the small scale, we would be doing dialysis in dialysis bags, which could take up to 3 to 4 days. This allows us to generate, and that was again limited to 1 gram, this allows us to generate kilogram batches in 2 days.
Wow. We're going to talk to Steve about some of the aspects of the bioprinters. We must be able to commercialise and complete the whole package in order to address the clinical challenges that are in front of us. Hi, Steve.
Good afternoon.
So Steve is heading up our additive fabrication capabilities here at TRICEP, and he's going to introduce us to some of our capabilities. It's a capability that we like to call printing printers. So tell us about that, Steve.
So we've been very fortunate. We have a range of additive fabrication tools, and we've recently been able to expand on that set of tools. This is just one of the new printers we have. It's a Mamaki 3D printer, allowing us to print components in full color representations. And that's now allowing us to give much higher level of details to our collaborators and our commercial partners to get real realizations of the parts that they want to be able to produce. These systems are then enabling us to print new components for new printing systems, and we'll be able to run you through a couple of those systems right now.
Of course, all of these projects we're going to look at are driven by real clinical need. I mean, we're very fortunate, aren't we, to be able to work with clinicians all around the country who are really defining the challenge with us, defining the plan and implementing the plan, and then helping us in terms of translation.
That's the real driver behind what we're doing, and it's giving us great encouragement to work outside of just doing research for the sake of research and being able to treat real clinical needs. And this is one such example. We're referring to this as the iFix pen. This is a device that allows us to deliver media or deliver two materials straight to a corneal ulceration in the eye, allowing us to treat that corneal ulceration, and in either the domestic environment or an overseas environment to be able to limit the chance of infection and ingress into the eye.
So the bio-ink is obviously an important part of this, and that ink has been designed to be compatible with the eye, to be transparent and to facilitate the healing process. And then I guess that the hardware or the iFix pen was designed around the requirements to deliver that bio-ink.
That's exactly correct. And the reason why this is, we always see these delivery mechanisms as tools. They're allowing us to take that fundamental material, the critical therapeutic material, and deposit it in the structure that's required. And this is one primary example where we're taking two materials, mixing them in a custom tip, allowing those two materials to cross-link and gel and be able to adhere onto the eye surface.
And it's been great to work with the clinicians as well, just on practical aspects of the ergonomics, how
of the
you hold it,
device.
how you deliver it.
And I'm probably holding it incorrectly as we
Yeah, I
look
don't
at
know.
it. I believe it's to be held in that manner.
Right.
But this allows us to refine that. And with the set of tools that we have here, it's very easy to go into an iterative process to refine the structure, refine the ergonomics of the structure so that they have better use of it in practice.
And I guess another thing about TRICEP is that we'll go that step further. Not only will we develop the prototype, but we will make sure that that prototype is ready for manufacture by working with commercial partners to do
Again,
that.
that's a huge advantage of this facility, and building on our existing commercial relationships through linkage projects that we've had in operation for many years, and defining what really can be manufactured at scale.
Yeah.
So we have, obviously, as we're designing components, we have to keep in mind design for manufacture rules and larger scale injection molding rules, which would be the typical manufacturing processes that we use. But also being able to take our customized prototypes and put them into more complex stages. So the example case of a handheld device is ideal for a practical use in a clinical environment, but for us to characterize that device, to have fine control of the device as we're going through our experimental protocols, it's ideal to be able to transfer and mount the system into a 3-axis stage or even a multi-axis stage to allow us to have fine control over the scaffolding structures that we produce, to then be able to determine the repeatability and the consistency of the printing system that we've developed.
Yeah, brilliant. Well, that's brilliant. I mean, so there must be a whole suite of these that we're involved in developing now.
There is now. So the iFix pen is one of the earlier, simpler examples where we've got two materials that are being extruded simultaneously and mixing at the tip. Another example case, which has a slightly higher level of complexity, is the Excelda pen. And the Excelda pen...
This used to be the BioPen.
That is correct.
Right. Excelda sounds much better.
It does. It's undergoing a rebranding. So with the Excelda pen, we have a slightly more complex extrusion geometry. This is a coaxial extrusion geometry, where we would have an inner core and an outer sheeting of material, allowing us to maintain cells within the inner core and then have a protective outer sheeting during the actual extrusion process. Obviously, this tip is then a bit more complex, and through some of the technology that we have here on site, we've been able to develop a robust, refined structure with a complex internal geometry that's been produced out of titanium alloy. So that's easily cleanable, easily sterilizable, and reusable in our preclinical trials.
So how did you make that?
So the design was done in-house. We optimized the internal fluidic pathway through a CFD analysis, and we were then able to produce it using metal additive fabrication. So the actual process is referred to as selective laser melting, allowing us to produce features with a minimum wall thickness in the order of 200 microns.
So it's printed on a metal printer, a 3D metal printer.
That's correct. So a 3D metal printer, we're very fortunate to have two of these systems available to us. The first is would have a cylindrical build volume, a 70 millimeter diameter tray with a maximum build height of 74 millimeters. The newest, most recent system that we've added to the facility is a concept laser 200R. That allows us to have a print volume of 100 millimeters by 100 millimeters by 100 millimeters. So a liter of volume that we can print within. We can print in a range of different metals. Stainless steel and titanium would be two of the most common materials, allowing us to produce high resolution, low surface roughness components that are ideal for customized prototypes.
So some of these structures must be really difficult to make at all using conventional manufacturing approaches.
Well, that's absolutely correct as well. So what you can do is, if you actually look at the supporting structure, and this is sacrificial supporting structure, we're able to make these intricate geometries, open porous lattices that in our mind are ideal for complex electrode structures, or indeed the production of complex fluidic geometries.
Yeah.
Okay.
But that's amazing. I mean, so you really are printing printers.
Well, we're moving on so that we, instead of having our handheld devices and integrating those into commercial type platforms, we're actually developing our own customized platform that's to allow us to have additional functionality to incorporate additional extruder or deposition techniques simultaneously, as well as cross-linking methods. So in this example case, the 3D ED, we're able to have two materials being simultaneously coaxially extruded with temperature management on both of those materials, and also the feature or the capability to be able to cross-link those materials as they're being deposited.
And this educational printer is an important part of our online teaching program that we're putting together. As you know, we have a graduate certificate in biofabrication, which is available online now. And that is actually the coursework or that's necessary to move on to the Masters in Biofabrication. And it's exposure to all of these types of printers that really give high-level, state-of-the-art training in 3D printing, isn't it?
That's right. And within this small package, our prospective young engineers and bioengineers have a range of tools, a range of capabilities for them to learn. So from the fundamentals of the 3D positioning systems right through to the development of the manual mechanical extrusion mechanisms, temperature management, to allow us to see what happens to our different materials under different temperature conditions, but also under different printing conditions or extrusion diameters.
And of course, building the printers is in itself a great dimension to the training, isn't it? We had a number of summer scholars working here with the team at TRICEP who actually were the first, I guess, to build the 3D Ed, and we're learning from that experience and how we can use that building of printers as a training aspect.
Yeah, and it's been a training session for us as well. Those students were able to identify the benefits for them in what they were able to grasp from our initial developments and what they were able to contribute towards it, so to how we could refine the design to improve our design of manufacture and design of assembly.
Great. So thanks for giving us that amazing insight into how we're using conventional 3D printers to create a range of customized printers, which is helping us to address important research challenges in the medical sphere, but also helping us to build new tools for training the next generation of biofabricators. Thanks,
Thank you.
Brilliant. Thank you. Hi, Zia.
Hi, Golden.
How are you doing?
Yeah, I'm great.
Good. So I've just been talking to Steve about all of this range of customized printers that we're making for our clinical collaborators. What's the clinical project that you're involved in?
This one is for the pancreas islet transplantation project. And this is the printer we developed for this project. We call it 3D PICT. It's the pancreas islet cell transplantation device. And in this device, we contain the 3-dimension printer with the core and shell structure. And this is some sample print in the core shell structure.
So the core and the shell, in this case, Steve was telling us about putting the cells in the middle of the core. What's the idea here? I remember that we need to integrate 3 different types of cells for the islet cell transplantation. So how would those 3 types of cells be integrated?
Yes, we encapsulate the pancreas islet cells in the core structure, which can provide more immunoprotection for the islet. And we can put the supporting cells, such like endocellular progenitor cells, and in the shell structure, and these cells will promote the vascularization process of the whole implant construct.
So
in the meantime, we also have this droplet device. They can drop the cells into the printed construct. So we have the potential to deliver 3 types of cells together into one construct, such as T-Rex cells, and these cells can protect the islet from the immunoprotection from the patient itself.
So the islet cells, they're obviously the key cell that the clinician wants to transplant into the patient. So they come from a donor, and they are going to respond to glucose and release insulin. And then you mentioned the endothelial progenitor cells, so they will help with vascularization, is that correct?
Yes. Yeah.
And then you mentioned T regulatory cells. Now,
Yes.
these T regulatory cells, as I understand it, they help to regulate the immune response, is that correct?
Yes.
Right. So this printer then can deliver all 3 types of those cells discretely in a 3D structure.
Yes.
Can it also spatially distribute them where you want them in order to optimize performance?
Yes, correctly. We can control the position of each type of cells. And for example, we can put the T-rex cells in the surface, in the outer layer of the construct, and protect the islet cells inside the construct. And in the meantime, the EPC cells will go through whole construct and promote the vascularization of whole construct.
Great. And I know that this project is carried out very much in association with Professor Toby Coates' group in Adelaide. So how does that work? I mean, do they now have a printer in their laboratory?
We deliver a customer-made, is nearly identical to this one, to their lab. And they also work hardly on that device and try to print the construct for animal work. We already implant some construct in mice, then we are targeted to the large animal samples for such as pig. Yeah.
Right. So it must be exciting to be involved in a project where there's very fundamental advances in materials, fundamental advances in how we understand the cells to work in combination with those materials, and then actually delivering those ink formulations in the hardware into a clinical environment where we can envisage translation of this technology to help individuals. That must be exciting.
Yeah, we are really excited to get involved in this project. We are not using our knowledge in the material and the biology and engineer and to develop the device and formulate the bio-ink formulation, and which can be used in the clinical and have potential to treat type 1 diabetes.
Brilliant. That's great. Thanks, yeah. Thanks
you.
for providing those insights for us. Thank you. Well, we have one last customised printer to look at. So I'm going to introduce you now to Johnson. Hey, Johnson.
Hello.
How's it going?
Good. How are you?
And our great friend 3D Ali.
Yes, this is the 3D.
What's Ali all about?
So this is a printer that we built for the clinical collaborator at Sydney RPA Hospital. So all of the components were built in-house, and this printer is able to print 3 different materials, each having their individual printing speed and conditions. So we sort of tailor them to be able to print different materials that require different printing parameters.
So like all of our other customised printer projects, of course, driven by a real clinical need and usually always actually by a passionate clinical collaborator. So tell us a little bit about our passionate clinical collaborator on this one.
So this printer was designed for the project where we regenerate the cartilage of the ear. So it's in patients with what's called microtia, and that's in the normal development of the normal ear. So what happens is that the ears, when the child is through the growing stage, they will have a deformed ear where they only have a little bit what's called a peanut shape ear. So it's different from a normal size ear, and the current techniques to treat that requires a lot of surgical process and leads to a lot of complications. So the passionate clinical collaborator came to us looking for a bioprinting solution. That's where we came up with this idea to print the entire ear out of biomaterials.
Right.
Yeah.
And so. And of course, that passionate clinical collaborator is Pal Mukherjee from the Royal Prince Alfred Hospital. So this is obviously quite complex, right? Because you need I'm guessing, a material that's going to retain structure, mechanical integrity, and another material for growing cells, and in your case, cartilage. So I guess that's why we have this multi-head printer that's been necessary for this project.
Yes, as you can imagine, if we were to just print an entire ear scaffold out of a gel-based material, it's not going to hold shape. And the ear is all about the aesthetics. So that's why we needed materials that give it the structural support, materials that give it the cell support, and then obviously a sacrificial material which is required for all 3D printing applications. That's why we have this complicated printer where we have 3 heads, one that cools down, 2 of them that cools down, and 1 of them that heats up the material to allow us to do melt extruding.
And so we've developed this with our clinical collaborator who's helped us to articulate the challenge and then develop the plan and put that plan in place. Where are we now in terms of transferring this into the clinical research environment?
So as you can see with some of these scaffolds, they come with different pore sizes, different orientations, and each of these different patterns correspond to a specific stiffness and flexibility, just like the normal ear, what we experience in the normal ear. So this is sort of the first phase of a project where we try to represent each of these regions by changing the parameters of the printing process. Then the second stage is then to incorporate cells in there and induce chondrogenesis, and which is what we're trying to do right now, inducing chondrogenesis in each of these different patterns and hopefully each of these regions because of the different stiffnesses and mechanical properties they would produce allow these chondrocytes to excrete out different amounts of matrices.
So obviously a very multidisciplinary project, bringing together material scientists like we've met today, and process engineers as we've met today, and mechatronic engineers and biologists and biomaterial scientists. I'd imagine that projects like this are both challenging but extremely rewarding. More challenging or more rewarding?
I would say a bit of both. It's definitely very challenging as the ear isn't one uniform stiffness as we can see. It's a whole range, and then we have to make sure select inks that are not only printable but also compatible with the cells, and that's sort of the challenging bit. The rewarding bit is once we can introduce this new concept, then we sort of solve a lot of the issues that we have currently in surgery where they have donor side mobility. And so this is a lot easier in terms of the whole printing process.
Well, we've had an exciting journey around our labs today, starting with the fundamental advances in material science and how we're using chemistries to retain all of those amazing properties we discover sometimes in the nano world of materials, and bring those through into processing structures like the sutrode and making devices like for the ear and the cartilage regeneration, and blending together all of the skills necessary to do that. And of course, working very closely with all of our collaborators who are real end users. And here at TRICEP, being able to complete that pipeline to take everything to that manufacturing level, the bioink that can be manufactured, the printer that can be manufactured, and all of those solutions delivered to our end users. It must be a very interesting experience to be involved in here at TRICEP.
It is. Over here, you can actually put your projects, the research project, into practicality, into a clinical scenario, and that's what's exciting.
That's what we're here for.
Yes.
Cheers.
So, yeah.
Well, we hope you enjoyed your tour of IPRI, the headquarters of ACES, and the introduction to TRICEP, where we're making things translatable so that we can solve real applications using fundamental advances in material science and engineering. If you have any further questions, please don't hesitate to contact us or hang on and ask us now. Thank you.